10,000 years ago, the average human life lasted just over 30 years. And then 100 years ago, that number was up to 50. And if you were born in the last few decades in the developed world, then your life expectancy is 80 years. But that is, of course, assuming that no major breakthroughs happen during your lifetime that can slow the process of aging. And that may be a very bad assumption. There's a new series on National Geographic Channel which was developed with GE, and the show's creators wanted to present my take on aging. So I'm here at the GE Global Research Center to talk to principal scientist Dr. Fiona Ginty.
And this would be kind of an example of, one of the types of images we would generate. So
what are we looking at here?
So these are cells that are actively dividing, and these are cells that are heading on their way to death or apoptosis.
Aging is not recognized as a disease. I mean, there are plenty of diseases we do acknowledge, like diabetes, heart disease, Alzheimer's, and at their core, aging may be responsible for all of them. And yet aging seems natural because it's something that we do from birth. And for a while it makes us better, bigger, stronger, faster, more intelligent. But then at some point in your life, it reverses, and aging makes our bodies decay and degrade. Why is that? Why do we have to age? Why do our bodies have to decay? Well, scientists are now realizing there is a fundamental cellular mechanism at the heart of aging. Do we age at the macroscopic level because our cells are aging at the microscopic level?
To a great extent, yes. There's only a finite number of times a cell will divide.
A key discovery was made by a biologist named Hayflick. He was studying normal human cells, and what he found was they can only divide a finite number of times. On average, it's about 50. Beyond that, the cell becomes senescent, which means it's an aged cell. It can divide no longer. It lives for a little while, but it's the accumulation of these senescent cells in our bodies that leads to aging on the macroscopic scale. So it's as though cells have this little timer inside them that tells them when to stop dividing. But how do they know? And what is that timer?
So telomeres are like like your shoelaces have, a little bit of plastic at the end to stop them from fraying. So telomeres are like the ends of your shoelaces.
But for chromosomes.
But for chromosomes. So they keep the chromosome together and they stop it sticking to other chromosomes. And so every time a cell divides, it loses some of the telomere. They estimate about 200 base pairs.
Why is that? Why can't it just copy to the end?
It's just sort of really the mechanics of it. There's only so much space when the DNA polymerase does its job of replicating, yeah.
So the telomere, and the telomere getting shorter is like your molecular clock, the cellular clock inside each cell that tells it how many times it has divided. Would you want to have your telomeres measured?
Well, people do get their telomeres measured. There have been associations made with lifestyle, with exercise, showing that longer telomeres are associated with, a more active lifestyle, exercise.
What if there was a way to stop the telomeres from shortening? If we could do that, maybe the cells would live forever.
In the early embryo, very soon after fertilization, there are stem cells that are able to differentiate or mature into any other tissue in our body. Almost all our organs contain a small population of these tissue-specific stem cells. In every tissue, many cells die when they reach their normal lifespan or after injury. Skin wounds recover very quickly in young infants. As we grow older, healing gets more difficult. Many old people suffer from too few blood cells and become susceptible to infections or anemic. It seems very plausible that at least some aspects of reduced tissue functioning during aging results from a reduced functioning of stem cells. How could stem cells age? As stem cells divide very often during the lifetime of an organism, they need to replicate their DNA and pass this on to their two daughter cells. Damaged DNA is therefore bad for stem cells. Special case of DNA damage relates to the ends of chromosomes, also referred to as telomeres. These ends shorten a little bit with every cell division. If the ends become too short, a stem cell cannot properly divide any longer. If we would be able to find ways to keep stem cells functioning properly during aging, tissues would remain more healthy.
As we age, our cells' telomeres gradually shrink or get damaged, making it harder for them to multiply. But by adding new stem cells into our bodies, we can help refresh and revive ourselves. This cool method swaps out old or damaged stem cells with fresh ones, kickstarting a renewal process that fixes telomeres and makes stem cells work better again. This helps fight the effects of aging and keeps us young.